Good afternoon, everyone. My name is Ritambhara Singh, and I'm going to talk about an interesting line of inquiry in our lab, which is integrating single-cell data sets using optimal transport. But before I start, since I'm attending this event for the very first time, a little bit about myself. So, my research lab focuses on developing machine learning and computational models with a lot of emphasis on data-aware and interpretable integration across modalities, whether it be in genomics or clinical or across the two domains. I did my undergrad in computer engineering from India and then my PhD from University of Virginia in computer science. And before I started at Brown, I did a postdoc at University of Washington. And today I'm not going to talk about deep learning at all, but we do do a lot of research in deep learning, and I also teach deep learning related courses. So if you want to discuss deep learning related projects, I can do that as well. All right, so moving on to the project, the motivation behind the whole idea comes from the fact that the advances in sequencing technologies have led to us being able to measure all sorts of different genomic phenomena. So we can measure how the DNA is arranged inside the cell in the 3D space. We can measure which parts of the DNA are open or accessible, that's known as chromatin accessibility. We can measure what kind of chemical modifications are taking place, like DNA methylation, transcription factor binding events, so these are proteins that bind to the DNA and start the transcription. And last but not the least, gene expression, whether the gene is expressed or not. And all these advancements have led to the exciting area of multi-omic study, where the idea is that we want to jointly study all these different measurements or modalities together so that we can get a comprehensive idea of what is happening when a cell develops or what is happening when a cell enters a disease state. Adding to this excitement are the advances in single-cell sequencing technology. So up till a decade back or so, we could only measure things in bulk. So the idea is that you take a population of cells, you do your experiments, and then you get some sort of an average signal about the genomic phenomena. But more recently, we can actually do these measurements at single-cell resolution. So you can imagine, since we can measure things at single-cell level, it reveals more heterogeneity that may exist in our data sets. A very popular example that people use in this field is that of a smoothie. So you can think of bulk sequencing as a smoothie, so you have a general idea of what's happening. But what single-cell sequencing allows you to do is look at each individual fruit that went into your smoothie. However, trying to do multi-omic study in case of single-cell sequencing technology is very challenging. It's challenging because once you take one measurement from your single cell, the cell is destroyed, and so you don't have any material to work with to take the next measurement from the same individual cell. However, there are some experiments that exist that allow you to take two different measurements from the same cell. These are known as Coassay experiments, and the idea is that they look at different molecules within the single cell and measure them or sequence them separately. While this may solve some of the multi-omic study related challenge, it turns out that there aren't that many Coassay data sets to begin with, and that's mostly because a lot of measurements require access to the same molecule, which is very hard to do at single cell level, and B, that if you were to study any new combination of modalities in the single cell, you would have to develop a new protocol, which is not only difficult, but these protocols tend to be very expensive. So how do biologists end up doing multi-omic studies? Well, the way this works is then now we sample from a cell population, we do measurement 1, and then we sample from the same population and we do measurement 2, and now we have two measurements coming from roughly the same population. And so what I'm showing here is we have taken gene expression data from a population, we get these matrices, cells by genes, and what I have done is I've plotted it into a 2D space using PCA. And the same thing for chromatin accessibility. And you can see that it looks pretty nice. There are some clusters emerging in your data. Each point is a cell. The challenge comes when I try to start integrating this information, because if I don't have any prior knowledge, I don't know if this cluster corresponds to this cluster across the two data sets. And that's because I don't have any corresponding information between the cells because we didn't measure the same individual cell. And because we measured two different measurements, we don't have one-to-one correspondence between the features. And so developing unsupervised data integration methods is very important in the field. Now, there is a whole list of methods that do this single cell data integration, but a lot of them require some notion of corresponding information to be able to do that. What we are trying to tackle is a much more challenging task where we are saying that we don't have any information whatsoever, and so we want to do this in a completely unsupervised settings. So, yes.
So why do you want to do this? Which question do you want to answer? Like an example of a question that you want to answer by combining.
Yes, so suppose I have taken gene expression data and I have taken chromatin accessibility data. You might want to say that, oh, is this cluster that I'm seeing, this cluster of cells, is it the same with that cluster of cell that has a certain type of gene expression? So that we can do some sort of matching, like what's the gene expression profile and what's the corresponding chromatin accessibility profile for the cells that have been matched. Mm-hmm. All right, so unsupervised algorithms, we started developing them in 2019. The first two methods that came out were MMDMA and UnionCOMB. MMDMA is a method that I worked on during my postdoc. These methods showed promising results, but like any other computational methods, they had hyperparameters. So hyperparameters are parameters that are associated with your algorithm that the user gets to pick for different algorithms to work and get optimal results. And so if you don't have any corresponding information whatsoever, you don't have a validation data to pick these hyperparameters optimally. So we came up with the idea of trying to do this alignment using optimal transport. And we didn't know at that time, but what we found out was this method allowed us to develop heuristics so that we could actually self-tune the hyperparameters. And I'll talk about that in a bit. Soon after Scot came out, a few more unsupervised methods came out. And again, they all showed pretty good results, but a lot of them were tested or benchmarked on Coassay data sets. You can imagine you are measuring two things from the same cell, so you have one-to-one correspondence that you can use as ground truth to test your method. But it turns out that there are a lot of challenges associated with non-Coassay data sets, which are actually the real world use case for these methods. And all these methods, including our own, Scot, struggled with these challenges. And so we came up with a new version of Scot, which was able to handle some of these challenges. The main challenge being that it was able to handle disproportionate cell type representations across the two data sets. So you're sampling from a population, you cannot guarantee that every time a cell type will be represented in the same proportion. So we were able to handle that, and we were also able to integrate more than two data modalities. And then more recently, we have been asking ourselves these questions that most methods can apply cells, but can we also get some information about how the features are aligning? And this is interesting because it can allow us to develop some interesting hypotheses by looking at what features are mapping across the two data sets. So the most recent method we have developed, it's called Scooter. And what it does is that it jointly aligns cells and features. What that allows is it improves cell alignments and it yields hypotheses on the feature mappings. And another thing that it allows for is weak supervision. So in the interest of time, I'm going to focus on Scot because it will lay the groundwork, and then talk about our more recent method, Scooter. If you're interested in learning more about Scot version 2, I'm happy to talk about it offline. So Scot was led by the development was led by two PhD students, Pinar and Rebecca. And the whole idea was that we have two data sets coming from two measurements from cells. And what we did was we said, let's treat these measurements as probability distributions. And once we did that, we could apply optimal transport to match the two distributions. So the whole idea behind optimal transport is we are trying to learn a probabilistic coupling matrix or a correspondence matrix, which are just weights, such that when we weigh one distribution using those weights, it maps to the other distribution. And the way we do that is by optimizing this optimal transport function, where we are basically trying to learn or minimize the probabilistic coupling matrix, which is the weight such that when we weigh one sample, it gets moved to the other sample in the other distribution. And we do that by minimizing a cost function that we define over the samples in the two distribution. Now, this formulation exactly won't work for our case because our two data sets are in two different metric spaces because they are two different measurements. So, what we used was a variation known as Gromov-Wasserstein optimal transport. And here we are still learning the probabilistic coupling matrix, but the cost function is not defined over individual samples, but pairwise distances. So, we calculate pairwise distances within the two data sets and then we try to map those. A very high level intuition here is that when we are mapping one data set to the other, we are making sure that the underlying structure, the topology is preserved when we do the mapping. So, that's the overview. Let's look at the framework. So, we have two data sets, domain X, domain Y. We initialize all the marginals as uniform. So, in the beginning we are saying everything maps to everything uniformly. And because we need pairwise distances, we calculate these distance matrices. You can use anything you want. The thing that worked really well with us was calculating these distances using short path distances that we calculated by first creating KNN graphs. And then we feed this into the Gromov-Wasserstein function. And from this, we get our coupling matrix, which allows us to map distribution 1 onto the distribution 2. There's a lot happening in that equation, so let me walk you through it one by one. What we want to learn here are these coupling matrices. So, that's that. We are learning them by minimizing the pairwise distances that we have already calculated for the two data sets. And this term over here, it's a regularization term which is controlled by epsilon hyperparameter. And this basically lets us control how homogeneous we want our coupling matrix to be. So, if all the probabilities are very homogeneous, the entropy would be high and vice versa. So, once we have optimized this and gotten our coupling matrix, we can use something called barycentric projection to project one data set onto the other by weighing all the samples using the coupling matrix. We can also use this coupling matrix to project the two data sets in some latent space for better visualization if need be. So, benchmarking, we took two Coassay data sets, SCGEM and SNARE-seq. Again, these are Coassay, so two measurements taken from the same cell, gene expression and DNA methylation for SCGEM, and gene expression and chromatin accessibility for SNARE-seq. The points are the cells, the colors are representing different cell types. Now, because we have this information, we use it as ground truth. For validation, this information doesn't go into the algorithm in any way or form. So, we applied the Scot on these data sets, and since all the colors look consistent, it's good news. It means that the method is able to align the two data sets and such that the cell types are preserved. If we color it by domain, we see pretty decent alignment across the two data sets. We also quantified the performance using something called a Fox-TTM metric, which we developed during the MMDMA work. Just intuitively, it captures alignment error, so lower score is better. And what we found was when all the hyperparameter tuning was done and we picked the best hyperparameters, Scot was able to give comparable performance to the state of the art at that time. Which brings me to this idea about the hyperparameters. So, Scot has two hyperparameters. One is the epsilon, that's the term associated with the regularization term. And the other one is K, which is associated with the KNN graph construction. Then MMDMA has 3, UnionCom has 4. And you can imagine that if you are working with a completely unsupervised setting, that you have no idea about what needs to match to what, it's very difficult to pick a good set of hyperparameters. Now, lucky for us, what we discovered when we were doing all sorts of experiment was that the Gromov-Wasserstein distance term corresponded pretty well to the alignment quality. And what I mean by that is whenever we saw that that distance was very, very small, it usually meant that the alignment error was also small. It did not always lead us to perfect alignments, but it still led us to reasonable alignments. And so we said that, well, if there is no validation data, no prior knowledge whatsoever, we can basically use this as a proxy to pick a reasonable set of hyperparameters. And so we tested that. So these are the results with the hyperparameter tuning. If we were to do this in a completely unsupervised setting, the performance obviously became worse for all the methods, but we were able to actually pick some reasonable set of hyperparameters that give some decent alignment performance compared to the other two methods that had no way of doing it. So we picked default values for them. So it's an unsupervised method. It can align data sets well. It has fewer hyperparameters, and the best part is that it has a heuristic to pick the hyperparameters when you don't have sufficient data. As I already mentioned, there's a better Scot version 2 out there, which is able to handle the disproportionate cell type representations in non-Coassay data sets, as well as combine multiple modalities. The code for both of these methods is on our GitHub site. So then moving on to the story for Scooter. So this work was led by my PhD student Pinar and our collaborators Hue and Yevgen. So Hue and Yevgen have been working for a while on this co-optimal transport formulation, which is basically jointly aligning samples and features. And we discussed with them that it might be an interesting idea to try those methods on single cell data sets, which we did. And we presented our earlier formulation, which is on bioRxiv at different venues. And based on the feedback we got from those venues, we actually updated the method to work better than what we had before. And that is what we called Scooter. And the change that we made was we added this gromov-cysteine term to it. That's why it's fused gromov. And I'll talk about it in a bit. So again, we have two data sets. For gene expression, you have the cells and the features are the different genes. For chromatin accessibility, you have the cells and then you have different chromatin regions. Now, most methods would be able to align the cells for you and tell you which cells map to which. But there might be a lot of interesting biology to be learned if you could get some sort of a mapping on the features, telling you what genes align to what kind of chromatin regions when you combine these two data sets. And so this is the task that we are trying to solve with Scooter. And we go about it by keeping our Scot formulation. So we are still doing everything that we did for Scot, and we learn a coupling matrix for the samples. The change we make here is that we add another term which comes from the co-optimal transport formulation, where now instead of just learning one coupling matrix for the samples, we learn another coupling matrix which is for the features. And we weigh these two terms by using the alpha hyperparameter. And then we have some regularization term. We still keep the entropic regularization term that I already discussed. And then the scale divergence term is something we introduced in Scot version 2 to handle the disproportionate cell type representation. So Scooter solves for two coupling matrices through an alternating optimization strategy. One is learning couplings between the samples. The other is learning couplings between the features. So once again, we start everything uniformly. We have the optimization strategy that until convergence first optimizes the cell alignments and then it optimizes the feature alignments. How does it optimize the cell alignment? Using this function. If you look carefully, the cost matrix is being computed by using two terms. The first term is exactly the same that we saw in Scot. The second term is basically we are calculating a cost matrix when the features are aligned using the coupling matrix in the previous iteration. So if we are starting from scratch, it's a uniformly distributed matrix, but as the iterations go on, we keep adding that information. Once we have this cost matrix, we optimize for the sample couplings over here. Now once we have optimized for sample couplings, we fix this and use it for learning feature coupling. So now our cost matrix is defined over the features such that we learn it by aligning the data sets using this coupling matrix that we learned for the samples, and then we use this cost matrix to optimize our couplings over the features. There are two advantages of this formulation. By adding this term, we are ensuring that the underlying geometry or the topology remains consistent when we are matching two different data sets. And this formulation where we are learning both samples and feature coupling gives us this feature mappings that allow us to develop interesting hypotheses. There's actually a third advantage of this formulation, and that is that we can allow for some weak supervision either on the sample level or the feature level if we have any prior information. So suppose this is the cost matrix that your algorithm computed for the samples. Now suppose if you have some prior information that these subset of cells should match to these subset of cells, then you can define a supervision matrix where you give lower scores where the mapping is correct and higher scores where the mapping is incorrect, and we can basically scale our cost function, a cost matrix, so that the algorithm has some constraints on what scores it should change and what scores it should not. And so because we are doing this in an alternating way, we can, if we have prior information on the samples, we can give that supervision at the sample level. And if we have some prior information on the features, we can give that supervision on the feature level to improve the other levels alignment. Yes?
One question for the supervision, what type of information will you be providing? So, for example, for the samples will be like one marker gene for the cell type, and that's the information that you are using. But then for the features, what type of supervision will you be providing?
Right. So I have an example coming up in 2 slides, so that might clear this question. It's very hard to define. I think it would vary from task to task. So we have one example to show what that could look like, but we can talk about other possibilities. Yeah. Okay. So benchmarking. To make sure that we didn't break anything with this new formulation, we tested it on our SC-GEM and SNARE-seq dataset. What we added to this was the site-seq dataset, which is again a Coassay which has gene expression and antibody abundance. And so we did we made sure that the cell-to-cell mapping was working with Scooter. The other nice thing about site-seq is that you have some notion of ground truth on the feature mapping, that the antibodies should map to their encoding genes. So we have some notion of ground truth there. And then we added another data set which will answer your question, which is the cross-species gene expression data set, which is gene expression on mice and lizard. And again, in this category also we have some notion on what cells should match and what features should map. So these 2 data sets were mostly added to see if our feature mappings made sense, because that was a very hard thing to try and validate. So cell mapping, whether the 2 different modalities map for the 3 different data set, it turns out that in unsupervised settings, Scooter is able to map these 2 data sets. We also quantified the performance, so it did better than Scot version 2, which was performing quite well to begin with. We also added this baseline called BindSC. BindSC is a very nice method, but they don't really technically produce feature mappings. But my student figured out that they do calculate it in some part of the code, so she was able to extract it. So we added this to just make things extra fair as far as feature mapping comparisons are concerned. So we tested how well the model was able to get the feature mappings for site-seq dataset. And so the 25 antibodies should be mapping to their encoding genes. And what we see with Scooter is this nice, strong diagonal. We also get a diagonal for BindSC, but you can see the weight is a little bit distributed across other genes and antibodies. And then the final experiment, and this was inspired by another collaboration that was happening in the lab, was trying to merge these 2 data sets for gene expression across 2 species. So here we had some notion of ground truth that certain cell types should be mapping to certain other cell types across the 2 species. But this was a very hard task to do, this mapping in an unsupervised setting. And so this is where we tested our weak supervision. So we said that the orthologous genes across the 2 species should match. And so we can provide some supervision on the feature level and see if our cell-cell alignment improves. And what we found was that with minimum amount of supervision, we were able to improve the alignment quality between the cells. And then we tested it the other way around, where we said, well, let's use some of the information we have about what cells should match, what cell types should match, and use that to see if we can improve the alignment of the orthologous genes. The alignment of the genes. And what we found was that, again, with a little bit of supervision, the gene alignment improved. Does that answer your question? That was one example that we had for samples and features. Okay. So yeah, so Scooter is able to jointly align cells and features, and that's interesting because the feature mapping can lead to all sorts of interesting hypothesis generation. The joint formulation improves upon the quality alignment that the quality of alignment that we saw for our previous methods. It also allows for supervision on either levels, which increases the alignment quality for tasks that are harder to do. This new formulation that I discussed today, it will be posted on arXiv soon, but we have the code available if people want to try and play with it. So this kind of wraps up the story of our attempt at single cell data integration using optimal transport. Big thanks to all the students who worked on this, especially Pinar, who this was basically her PhD thesis and she recently graduated. Rebecca, who was very instrumental in kickstarting this project in the beginning and helping us with some of our initial formulations, and Manav, who is an undergraduate research student in the lab who has helped us with running experiments. Thanks to our collaborators, Bjorn, Bill, Yevgen, and Hui. Thanks to all the members of our lab. It's been wonderful working on all sorts of different projects with these talented bunch of students. And thanks to my affiliations and funding sources, and I'm happy to take questions now.